Within the cell, all genetic information is stored within DNA. The genetic information is made functional by the process of transcription, where DNA is transcribed into RNA. Once transcribed, most RNA must be processed to become functional. This processing includes addition of a 5' cap, 3' polyadenylation, and splicing to remove intronic RNA. We will now explore the splicing process through dance. Following transcription, pre-mRNA must be spliced into its mature form. This is accomplished in the nucleus by the spliceosome. In this video, we see the upstream 5' and downstream 3' exons, the RNA sequences that are included in the final spliced product, and introns, which are removed and degraded. A major player in the intron is the attacking A, which is important for catalysis at the 5' exon-intron junction. We also see the additional splicing factors U1 and U2 small nuclear ribonucleoproteins, or SNRPs, complexes made of both RNA and proteins that both organize and catalyze the splicing events. In the first step of splicing, the U1 SNRP binds the 5' splice site at a consensus GU site, and the U2 SNRP recognizes and binds the intronic attacking A. These SNRP complexes bring the 5' splice site and the attacking A together. At this point, the 2' hydroxyl of the attacking A cleaves the phosphodiester linkage between the upstream exon and the intron, forming an intronic loop and leaving the end of the upstream exon with a free 3' hydroxyl group. This loop of intronic RNA is known as the lariat. To finish removing the intron, the 3' hydroxyl of the upstream exon is repositioned to attack the phosphodiester bond of the downstream intron-exon junction, connecting the 5' and 3' exons and releasing the lariat intron, which will be degraded. One of the most remarkable discoveries of the last century was the existence of enzymatically active RNA. Prior to this, the central dogma of biology held that DNA and RNA merely existed as the mechanisms of heredity, while proteins were the biocatalysts. The first of these ribozymes found was the tetrahymena thermophila self-splicing intron. As the name suggests, this RNA molecule is capable of excising itself from the pre-RNA and ligating the surrounding exons. In this reaction, the RNA features are identical to canonical splicing, with exons that will be incorporated into the final product and the intron that will be removed. Note that we no longer have our SNRPs, and importantly have gained the exogenous G, which will serve a similar function as the attacking A in canonical splicing. To begin, the intron folds to form the G binding site, and positions the G to attack the phosphodiester bond of the 5' exon-intron junction. This leaves a free 3' hydroxyl on the upstream exon, which is then repositioned to attack the phosphodiester bond at the downstream intron-exon junction, excising the linear intron and releasing the mature RNA. The intron then autocyclizes, releasing a short oligonucleotide and a ring of RNA containing the bulk of the intron, which is rapidly degraded in the cell.